on Canvas under modules. But it's pretty self-explanatory what's in the, it's pretty clear, thorough as well for how to do this, but I just want to go over a few things. That's it. Okay, so it's a journal assignment, essentially a scientific paper, a review paper that you're going to write on a certain topic. And I actually have a list of possible topics that you can write on, and I'll be posting that as well. And first thing I'll say is you don't have to choose one of the topics. If you come up with one that's not on the list, you can definitely use it. I would email me first to make sure, only because I've had people in the past write on something that's not related to biology, but more related to like chemistry or physics even, or, you know, something, yeah, like earthquakes, things like that. So it's not really, wasn't related to biology. So there's a list of really cool topics that you can choose from, or come up with one that you're interested in. Totally up to you. Okay. So you're going to be writing this paper, basically talking about summarizing a specific topic that in biology that you chose. I'm going to actually, oops, get something out here. Okay. You can work with a partner if you want, except none of you probably know each other because it's not in the classroom. So if you do, feel free. Feel free if your friend, you know, just keep in mind you'll get the same grade. So don't pick someone where you're doing all the work or whatever. Okay, let's see. So you're going to be essentially researching this topic on the internet, various websites, resources. There's so much out there, but you can't really use Wikipedia as one of your resources or to cite. At least one of them must be a primary source. And I have a link in the directions that will be on Canvas for what that means. It's basically primary source literature in science is peer-reviewed, published, credible, high-level scientific journal paper, and you only need one, at least one. I recommend going to the following site. Everything you'll find on this is primary source literature, but some of it, because we're not on MiraCosta's Wi-Fi and they usually give you access, you have to pay, but there's also a good amount that are free access. So if you do some digging, you'll definitely find a lot of them that are free to look at. But to start, you can go to www.pubmed.gov. And there's a search bar. You can type in your topic, and there'll be thousands and thousands of primary source literature, research papers that come up. So I have a little bit in the directions on format, font size, things like that. Something I will talk about first is what we call an abstract. So in the very MLA, actually, so the format is not MLA, it's, what is it? I forget. It's the medical one. APA for citations. Thank you. Yeah. So the first thing I'll mention is the abstract. So the abstract in science, and you'll find out an abstract on every single one of these papers. It's the very first thing you see. An abstract is not an introduction. It's not a preview or sort of, yeah, or an advertisement, or but mostly I've seen in the past people do the abstract as an introduction to your essay or your paper. It's not. What it is a summary. It's a summary of your entire paper. Really just key findings and facts that you learned. Abstracts are short. They're like a paragraph, like 5 sentences. But I'm going to be looking to see if you're introducing your paper, which I don't want. That's more of like English. English papers do that. An abstract is, I want to read your abstract and know exactly really the most important things about your paper. And then when I read your entire paper, I'm really just reading details and extra information, but I know the key crucial things that you discovered or you wrote about. So I like to mention that because a lot of people don't do that. But that, again, it's in the directions. So the information, you're going to be writing, how long does it have to be? I knew that was coming. Essentially, there's no like page limit. Okay, so, but I will say, you're going to have your main topic. And in fact, I have some examples I just pulled out. I had students in the past do DNA forensics as a topic. I've had people do bioluminescence and write about that. Gene editing. So essentially, when you choose your topic, you're going to have 4 to 6 subtopics. For each one, you're going to write 1 to 2 paragraphs of information. Again, this is all in the directions. But when you do this, when you write, maybe I should have sent you this beforehand so you could follow along. But I'm describing it. And it's all here, you'll see it. But for each subtopic, you want like 1 to 2 paragraphs. When you do that, if you have 4 to 6 subtopics with 1 to 2 paragraphs each, and there's illustrations that you're going to have, which I'll talk briefly about, it comes out to maybe like 6 to 8 pages in length. I always get that question, how many pages? So 6 to 8, probably there's nothing wrong with that question. I used to ask that question. 6 to 8 is probably the best. If you're at 6 to 8 and you follow all the directions, you're in good shape, I would say. Okay, speaking of subtopics, let me just give you some examples of what I mean by that. So if you chose DNA forensics, some great subtopics are the history and progression of DNA forensics. Another subtopic this student chose, forensics in criminal investigations. So examples, I suppose. Wrote 1 or 2 paragraphs on that. Had a subtopic, obstacles, DNA forensics. And his last subtopic was improving the technology. So those are great examples. I wonder if I don't have one for a disease, but usually subtopics, if you choose as your main topic a disease, there's usually a couple that are pretty good. So let me pull up one here from my past. Let me see. Oh, I think she did. Okay. This is a great one. Okay, so this student chose AIDS as her topic, and her subtopics were, the first one was how AIDS and HIV affects the body. The second subtopic was how AIDS is spread. She wrote 2 paragraphs on that. Genetic factors as a subtopic. History and impact of AIDS as a subtopic. And those were her 4. So 4 to 6, right? So those are some examples. symptoms, things like that. Those are great subtopics for something like if you choose a disease as your main topic that you're interested in looking at, like malaria or tuberculosis, or a lot. Okay, what's next? Illustrations. Oh, for writing. Oh, correct. So Emily asked, we start with the summary or the abstract, same thing. And then you go to the subtopics, exactly. So you'll have your abstract, and then you'll jump right into your first subtopic. Exactly. And your abstract really should probably have 5 sentences or so, with probably your most important sentence from each subtopic. That's a good way to do it. Okay, cool. Illustrations. So at least 3 figure captions with under each one that sort of explains what I'm looking at and why. Like why this is here, what's it telling me, that kind of thing. So that's here as well. I like to talk about in-text citations. So APA format, in-text citations, that means it's the author's last name, comma, the year it was published. So this is for a citation that's literature based. So like, so article or book would be in parentheses. So you have, and there's an example here. So I can actually show you guys. So here's an example of, so you would have a sentence like this. Over 400 major earthquakes occurred in this town or whatever, in whatever. So you research that, right? So where'd you find that information? In the text, in your paper, you would have the author's last name, which is Smith, and the publication date, year, 1990, just like that. You'll have those throughout your text. Doesn't need to be like after every sentence. Just, what is that for? So that's for basically giving credit to those who did the work or the research, or so you're not, it's sort of a practice that we do for copyright and for, you don't want to, so that people aren't assuming you're taking credit for something that you didn't discover. Obviously, this is an assignment in a class, but we want to practice these types of things in science. You never want to make a claim and then somebody else did the, for the subtopics. Yes. So Emily, yes. Throughout your paper, you'll have in-text citations. It is also proof that your sources are reliable and credible. Absolutely. So those are the in-text citations. And if it's a website and you can't find any sort of article or book or whatever on it, like for instance, your primary source will definitely be like that. Author and year, it'll be an article. But if it's a website, you do it like that. This is all in here, and I don't want to keep you guys too long. So it looks like that with the URL and the date that you accessed it, or that it was published, actually. Or if no date is available, you do the date you accessed it. Okay. You'll have a literature cited page at the end. Again, it says you have at least one primary source, journal, scientific journal article, APA citation style. There's a website for that, what that means. Grammar and spelling, it's important. It's part of the rubric. I just have a little blurb I took from someone on that. It's important. So make sure it's, have someone proofread for you. Let's see. Anything else? One thing I like to mention, don't plagiarize. I will find you because I do have Turnitin or whatever that software is. Miracosta's is actually integrated right into Canvas. So you want everything to be in your own words. And you also, right, so you don't want to be like copy pasting everything from other sources. You want it to be in your own words. You don't want to use quotations. In science, there's never really a need to quote anything. Just rewrite it in your own words. That shows me that you understand what it is saying. If you just kind of copy and paste it, a monkey can do that. No, not disrespecting monkeys. I think they're brilliant, beautiful animals, but you know what I mean. Anybody can just copy paste without knowing what the true meaning of what you copy pasted. So don't use quotations. I had a student last semester, her whole paper was quoted. And I was like, oh no. So don't do that. If something is so like incredibly complicated worded and you know what it means, but you can't really reword it, maybe one quote's fine. But just generally you want to avoid them. That's it, I think. I'll put the due date as well. Let's see. The due date was from the announcements. I'll email that out and it'll be in, it probably won't be in the directions, but I'll include it in the email for sure. Is there a packet this week? Yes, there's a lab packet this week. We're gonna talk about it next. Oh, okay. Well, anyways, so let me see. Anything else? That's it. Yeah, any questions? I talked about plagiarism. I talked about quoting, page length. I think I nailed all the main points. Should be fun. I like to do this as you get to look into something possibly interesting. The bioluminescence thing I think is cool because we just had that in San Diego. You can explore what it means, why it happens, why these creatures use it, things like that. So I hope it's fun. Anyways, all right, let's talk about the lab. We got a lab this week. Yes. So this week's lab is on pH, acid-base chemistry. It's a chemistry lab, pH and titration. So I sent you the packet. You should have it. Since we're talking about chemistry, this is right up our alley. We haven't gotten to pH and acidity and basicity yet, but we will on Thursday. But I'm going to explain the general concepts here so that Thursday's lecture will be more like reinforcing it. Okay, so a couple things we're going to talk about first. Okay. All right, first thing I want to just mention is what molarity means. What does molarity mean? So this is in your lab packet in the introduction. Molarity, otherwise known as, or abbreviated as this big M for molarity. Molarity is a measure of concentration of a solute in a solvent. So we haven't talked about that yet, but let's backtrack. A solution So a solution of substances in this liquid form is what we call when we combine something called a solvent and something called a solute. You get a solution. Okay. Now, what is a solvent? What is a solute? A solvent is the substance that the solute is dissolved into. It is the dissolving agent. So a solute is dissolved into the solvent, okay? So usually, I mean, always, the solvent, it's gonna be a large amount of liquid, and the solute is a smaller amount that gets dissolved in to the solvent. And together, these two liquids homogenize, and we call it a solution of two liquids. Water is the universal solvent of life. We're going to talk about that more on Thursday. But essentially, everything that is life is dissolved into water. All of the cells that make up me, you, all of life forms, all organisms, cells are 99% water. Cells are sacks of water with things dissolved in. Various carbohydrates, fats, and proteins, things like that. Okay. Solutes dissolved in. So when we talk about molarity, molarity is talking about the concentration of the solute in the solvent, in 1 liter of the solvent. So let's write that down. Molar concentration equals concentration, or how much solute in a solution, which is both together. Concentration of solute in a solution. So it's a measure of concentration. How much solute is in our solution that we're looking at? And if you have if you have increasing concentration of solute, you're gonna have increasing molarity and vice versa. So more specifically, molarity, we're gonna talk about what this means, is the number of moles. If you've taken chemistry, you're very familiar with what a mole is. I'm going to talk about it. Of solute, so moles of solute over 1 liter of solution. That's what molar concentration is referring to. The number of moles of solute, let's say it's 2, 3, 4, 0.05, whatever it is, in 1 liter, over a liter, per liter. If you have a liter of solution, it is the number of moles in that. Okay, what the hell is a mole? Let's talk about that. What is a mole? A mole is weird. It is this random number called Avogadro's number. We're getting a little bit into chemistry, so I'm going to kind of keep it at bio level. But 1 mole, it was a simplified thing. 1 mole is this crazy number, 6.02 times 10 to the 23rd. It's a massive number. This is 1 mole equals 6.02 times 10 to the 23rd, and we're either talking atoms, we could be talking ions, which we're going to be talking about today. Could be molecules. that kind of thing. It's a lot. 6.02, 10 to 23rd, that's like 6 trillion or something crazy. Much, even more. Billion has what, 9 zeros? So, a lot. One liter of the solution. Okay. Acids and bases. These are different compounds. Oh, boy, am I back? It says it glitched.